A very good morning to all the dignitaries, faculty members, academicians, and research scholars from the various institutes and universities. I, Dr. Bhavna Taneja, on behalf of Research and Development Cell, Amity University, Jharkhand, take this privilege of welcoming you to the fourth day of the FDP on the recent trends and challenges in research, organized by Amity Center of Excellence and Research under R&D Cell of AUJ. In the last 3 days, we have heard about the recent trends in the field of agriculture, science and technology, biotechnology, and today we will hear about the latest research in the field of humanities. I welcome the executive members of the FDP, Dr. Jayata Chattopadhyay ma'am, Assistant Director R&D, Amity University, Jharkhand, Dr. Soman Karnar sir, Associate Professor ASET, FDP coordinators Dr. Dhananjay Pandey sir and FDP coordinator Dr. Amit Kumar Datta sir and Dr. Shantani Banerjee ma'am. A very warm welcome to all the HOIs, HODs and my esteemed colleagues and the participants who have joined from all over India. It is my great privilege to formally introduce the first speaker of the fourth day of the FDP, Dr. Shrikant Rath. Dr. Shrikant is a chief scientist and professor of toxicology and experimental medicine at CSIR CDRI. He completed his PhD from BHU in 1995. He did his postdoctoral fellowship at CCMB with Lalji Singh Group, and he has been bestowed with a lot of awards. Some of them are the Raman Research Fellow Award, the Genomic Pioneer Award, the CSIR Technology Award for Innovation in 2009, again the CSIR Technology Award for Life Sciences in 2015, CSIR CDRI Incentive Award for Patents, High Impact Publications, Technology Transfers in almost 6 years, the Mridula Kamboj Award for Drug Development in 2021 and 22. Our guest, our esteemed professor, has produced 21 PhDs throughout, and he is a member of RCGM since 2014. He is a member of SEC DCGI from 2020 onward and is a member of DRC AIB Amity Lucknow 2018 onwards. A very warm welcome to you, sir. And we are waiting to hear from you, sir. So over to you, sir.
Already. Okay.
Dr. Shrikant Rath, sir. Over to you, sir.
Thank you, Dr. Bhavna. I am grateful to you all to receive this invitation to address some of the people who are participating in FDP. There is some technical glitch still Amit is unable to solve, perhaps because the slides he has not yet received. So I will again try to send it back. So good morning, everybody. I'm very, very sorry that this technical glitch actually due to the bad climate here in Lucknow. It is raining outside, so maybe that is the reason why the internet is very slow. But anyway, we are now coming back to the job. That is, I am going to speak about the development of a phytopharmaceutical opportunities and challenges. You must be knowing there is a new regulation in this country, which has actually they have started. CDSCO or DCGI has started very recently, that is in 2015 November, that we can develop phytopharmaceutical in this country, just like the botanicals in the United States of America, where they have FDA regulations for developing the plant origin products, unlike our Ayurveda or Ayush products. Okay, so the phytopharmaceuticals are nothing but the similar can you change the slides? Before going to the phytopharmaceuticals, let me define that pharmaceuticals are the substances that are used in the diagnosis, treatment, or prevention of diseases and for restoring, correcting, or modifying organic functions. Okay, so phytopharmaceuticals are nothing but, next slide, a similar type of pharmaceuticals which are actually originated from plant derived product, as we used it in Ayush or Ayurveda or homeopathy or in Siddha or Unani. So basically this is a scientific way of development of plant chemicals for use as a medicine. So as the name suggests, the phytopharmaceuticals are plant derived. For centuries, we used plant derived medicines. Ayurveda has started long back before the AD. So maybe around 3000 years back, we started Ayurveda. We have the Charak Samhita and Susu Samhita were well defined books. They are described about this all medicinal effects of different plants and how they are mix all these plant products and make the Ayurveda. And homeopathy is little better. In 15th century, Hanuman actually started doing the homeopathy practices, homeopathy drugs out of the plants just to avoid the uh, adverse effects of the plants. So Siddha is very unique drug treatment, in especially in Tamil Nadu. It is our own Indian origin, and a lot of mixtures and with all elements of different kinds mixed with the plant materials, and the Siddha practice has been there in the Tamil Nadu. These medicines are made from natural sources and have gained popularity in recent years due to their effectiveness and minimal side effects. I would, as a toxicologist, I will say that minimal or maximal side effects. That is why we are trying to do it in a phytopharmaceutical mode, so that scientifically we can establish the adverse effect, expected adverse effects, as well as the side effects. One of the main benefits of developing plant-based medicines is that people's acceptance is better, because people think that plant originated materials are better and acceptable, and they are taking every day different types of plants, fruits, leaves, roots, and barks. So the acceptance is certainly better than the synthetic molecules. Plant-based medicines uh, have been shown to have anti-inflammatory, antimicrobial, antioxidant, and different types of medicinal effects. As you must be knowing, there are medicines. For example, we have a very age-old drug like quinine. We have artemisinin. All these are plant-originated things. Then later, people have synthesized and semi-synthesized the products, and they are very popular in the market for different diseases. The success of NCs is now meager, because the other aspects of the things that for the last 30-40 years people have been trying their level best to find out some new medicines, new molecules for different diseases like cancer, microbial resistance, filaria, malaria, leishmania, but we are not very successful as it is expected. So basically we go up to the some stages of clinical trial, and the drugs are being failed because we are not getting the proper effects which are expected, because regulations are stringent now, and we have lot of screening tests where we can actually dissect the molecules effects in all different models. Therefore we are unable to actually pinpoint a good drug without any side effects or without any adverse effects. Next slide. Then, as we are failing in the new pharmaceutical molecules, we are actually trying to develop the phytopharmaceuticals, which may be a unique set of challenges, which may provide some unique set of challenges for researchers and pharmaceutical companies, because they are originated from plant, and isolating and identifying plants have different types of chemicals in its own leaf or its fruit, or in barks. So whatever material you are taking, you have to analyze them scientifically and identify all the chemical markers inside it, so that you can actually focus in what are the ingredients that are actually bringing the better effects or disease curing effects or diagnostic effects. That is why the challenges are very, very different. Then one major obstacle is variability of the plant-based medicines, because in different parts of this country or any part of the globe, if the same plant, actually they have different amounts of chemical materials, and according to the environment they change, according to the climate they will change, according to the soil conditions they will change, and according to the harvesting methods they can change. Therefore it is a greatest challenge. It may be very easy to talk about the that we can alternatively develop phytopharmaceutical, but it is certainly a very, very difficult task because of these obstacles. This can make it difficult to ensure consistent potency and purity of active ingredients. So that was one of the major factors. Scientific deliberations were made, and now we have some sort of guidelines in the new drug and cosmetics act 2019, which was published in March 2019. They have special definitions and special requirements for making phytopharmaceuticals. So lack of standardization in the production of the phytopharmaceutical is one of the major challenge, which we try now, people are trying to address it. And in my later slides I will show what we are trying here for development of phytopharmaceutical. Then unlike synthetic drugs which are manufactured under strict quality control standards, and we have to implement such standards in these plant-based medicines, which is actually little more difficult than the new chemical entities. This can lead to the inconsistency in the final product because of the, as we discussed already, that in climate changes, soil changes, or any place changes or harvesting methods changes, then there may be some changes and it is very difficult to assess them. Next slide. But there are a lot of opportunities. Opportunities is that we have a lot of medicinal plants, like 8,000 medicinal plants are already reported in India, and there are about 50,000 medicinal plants reported globally, and plenty of resources are there. And if you we try to explore only in initial stages, there are a lot of bottlenecks. But once people start phytopharmaceutical production, then of course it will be perhaps the fastest, perhaps we can produce them faster. Okay, so that is one of the opportunity. Then the phytopharmaceutical offer a natural plant-based alternatives, as I have already told, and they are totally strongly backed by the traditional knowledge. So we have a lot of traditional knowledge. You must be knowing, recently in Gujarat, the WHO has opened a very big center of traditional knowledge, just because India has a rich source of traditional knowledge, and to explore and exploit the traditional knowledge of this country, WHO is trying to make such investments in this country. So that will be a very good backup system for the phytopharmaceutical development. They can be made from the herbs and plants by standardization, and after through safety evaluations. Thorough safety evaluation has to be done, because scientifically otherwise people will not accept it. Then they can be safer and more sustainable than synthetic drugs, and that is possible because we have to get rid of the abnormalities, abnormal markers out of the plants after we analyze that they are the harmful materials which can actually affect the physiology of the human being. Some of the pharmaceutical originated from the plants have been shown to be highly effective in treating a wide range of medical conditions, and they contain, and in fact in FDA there are only two botanicals till now as approved, and both of them actually associated with the benefits to the patient who are suffering from AIDS. So there is a good competition opportunity because FDA is only after 23 years, they have only given approval to only two botanicals, whereas we have uh, in clinical phase 2, we have at least two molecules, two phytopharmaceuticals, which are in phase 1 clinical trial, and some of them are in the development process. They contain a variety of active compounds. Pharmaceuticals have a variety of active compounds that may work together to produce powerful therapeutic effects, which is not expected from the NCEs or new chemical entities, and maybe without the harsh side effects of associated with many synthetic drugs. So overall, there is a lot of opportunity in this area of drug development. Next slide. So this is a project, actually I will be talking about what we have done out of Ashwagandha variety, that is 118. It is a it's a old variety identified as Nimitli 118 because the project under Nimitli we have identified it. There are more than 100 types of varieties in this country. Ashwagandha, you must be knowing, it's a very popular drug in Ayurveda, and I think each and every households they have a ashwagandha tablet or powder packets where they are people are regularly taking. So that is what the thinking behind this so that this we can develop it as a phytopharmaceutical other than the Ayurveda. So that is what the basic thinking behind this uh, ashwagandha plant. Next slide. So these are the people who are actually my collaborator. I am also only a representative and coordinator, but all the people, there are more than 8 people. You can see 18 people who are working on it. There are many people who are technically supporting us, and I will be talking about how we have developed it up to what extent. Next. So there are objective is to develop a novel anti-stroke phytopharmaceutical formulation from the roots of an ashwagandha variety that is namely 118. It's a phytopharm. I am giving this example that through this I can show the path how phytopharmaceutical can be developed in this country. So basically it has to be developed like a new chemical entity, and it has to be filed as a IND, investigational new drug application, after completing the things. And exactly the same way we have to do the clinical trial as we do for the other chemical entities. Next slide. So the formula for any development of anything is actually NCs are basically you have to establish the activity, then you have to do pharmacokinetics, pharmacodynamics, availability in plasma, then making of material for clinical trials, then what are the excipients for compatibility and excipients for the formulation, then the toxicity and safety issues, which are very important. As it is a plant and we have phytopharmaceutical we are planning, so we have to also take care of the cultivation, extraction and fractionation and identification of this material. So here it is immediately 118 root, but any plant can be taken in that design. They have to be cultivated within a captive cultivation mode so that you can monitor what is the condition, what is the climate, what is the salinity, what is the condition of the soil, what is the water supply and everything, and what you are extracting, and from the extraction you have to do actually fractionation, because if you enrich the fraction then only you can see the accelerate the effects. Okay, the beneficial materials you can enrich in the extracts, then do the things so that you can actually get the benefit of a drug. Then you can find out the activity, of course, in a different models for different diseases. Different several models are there. So for this, as I have told, that is a stroke treatment. Actually, stroke, after stroke, what happens? The blood clots happens, and the brain actually divide of any oxygen supply. So when the oxygen supply is not there, what happens? The brain tissues are being damaged. So the main purpose of this material, what we are trying to develop as a phytopharmaceutical, is just to protect the neuronal setup, so that lot of damages should not be done. One. The other thing is, it is a ashwagandha which is very useful. So every household they have ashwagandha, most of the houses. So if there is any stroke anytime, immediately some ashwagandha powder can be treated. Ashwagandha phytopharma can be given, so that the neuronal protection will be there till the patient is going to the doctor. Because mostly it doesn't actually dissolve the clot, but it actually protects the neuronal side of the patient, so that brain at least is being preserved, so that lot of damages cannot be done. As we are developing it for stroke, we have to show this material that it is going to the brain. That's why we have done in the brain PK. And we also did it in the plasma, because one has to show whether it is a NCE or a phytopharma. One has to show that is actually going to the blood. So after reaching blood, it has to go to the brain, so that we can scientifically prove that it can be actually used as a phytopharma for the treatment of the stroke patients. Of course, GMP batches are required, because GMP is a good manufacturing practice. It is required because we are someday we have to treat this material for during the clinical trials, and we also did the toxicity and safety pharmacology studies. Next slide. So these are some of the pictures of our garden where we did the captive cultivation. These two left side pictures, left and center pictures, are actually in our CSIR CMAP lab, which is in Lucknow. Another as we have CDRI here, we have also CMAP, IITR, and NBRI. So CMAP have a lot of farms, and we did it captive cultivation there. And we also kept it in mind that once it is becoming a drug, the demand for the drug will be there. So the more demand for the drug, the more demand for the plant. So therefore, we also trained the farmers in Raebareli district, which is adjacent district to Lucknow. And this is the picture where farmer is standing in his field, and they are doing great in the current season, also 2023. These pictures are from 2022. Next. So as a requirement of phytopharmaceutical, as per the DCGI rules, that is NDCT rule 2019, we have to do the pharmacognosy to find out every type of nature and other things of the plant, and we have to do the transfer sections of the root, and so that actually, and it has to be identified by a botanist who is actually practicing the taxonomy. And the problem challenges here also, the lot of botanists, they do not know about the taxonomy, and you have to actually run to a known educated botanist who can actually practice taxonomy to identify your patent material, because that is the essential part of this phytopharma development. Then you have to grind and record all these things, and all these captive cultivations are to be done under the good agriculture practice. And good agriculture practice has to be, it is very difficult to followed by a farmer in the open country, but in the laboratories like us, we can actually do it, but it is really difficult for getting the farmers educated and advise them and monitor them there for captive cultivation. So that is another challenge we face in the making of this phytopharmaceutical. Next slide. So wherever you do, you have to actually analyze what are the things, where it is, what are the things you are physical parameters, heavy metals, microbial and aflatoxin content, because all these are toxicants should be avoided. And the DCGI ask all these questions, that whether you have analyzed all these things in the route where you have selected those roots you take out and dry them in the shed and wash them properly. After that, you take out the samples and check that whether they have the microbes or not, aflatoxins or not, or pesticides residues are there or not. You can see that all BDL are nothing but the below detection level, and all other where we have found the things, we have found what is the amount, and all these we actually follow according to the international rule, and which is acceptable. And this is where this is the chart of analysis in the roots what we used for developing this phytopharmaceutical. Next. So this is the fractionation. We did the alcoholic extraction, then did the fractionation by trituration, so that we can enrich the different materials like ethanoxide 4, 5, 6, ethaferin A, 12 DWL, DWL, and ethanolide A and ethanol. All these are actually active components of the ashwagandha root. So we found, you can find out in the 3 columns, the F1 has the maximum amount. So we actually showed that, proved that it is enriched properly after trituration. Next. So we all calculated and show this by HPLC methods, that how they are showing the, because these are the proofs they require in DCGI, and what are the molecules we are focusing on. You can see from this that in spite of these 6, there are many more peaks, and there may be some other chemicals we are not focusing, because we use these markers. We, out of these 6 markers, we will use in the future only 4 markers, which are consistently showing that they are not like getting degraded in the course of time. Next. So these are this is the place where you actually do the process batch up, scale up, and we compare them, and we can able to find out the comparability is almost comparable on the all these 5 materials markers in different batches, and we could able to show that we can do it further by in the lab in different batches. After that batch is we are confirmed that we actually shifted the forward, next slide, formula to the company, where we can actually get the batches in the similar conditions. So this is again the COA of this extracts, or this, sorry, extract, what I will say, this is the fraction, and you can see the materials actually where almost all, many materials are nil and others are reduced during the process of extraction. So we are actually purifying it further for a better phytopharmaceutical. Next slide. Then we made a from the company, and we got the similar results, and we could able to show that we can able to produce it in a company scale. More amount we can be produced in a company. A company can, if he is taking or she is taking, we can actually do the things properly, so that it can be done very nicely, and can be made as a drug. Next slide. Next slide. So these are all companies COH, where they have actually shown the things that all these materials can be identifiable as in the lab, and it can be uh, done in the in a company GMP environment. Next. This is again the COA of the GMP batches. Almost most of the materials are below detection level, because in GNP batches what happens actually, they have purification is more better than the laboratory scale. So naturally the materials are we are losing all dirty materials, so that's why they are most of the things are below detection level. Next. Then before making a formulation for treating the for treating the animals or animal models or human beings, we have to make a formulation. So before that, all the pharmaceutics people, they must be knowing that you have to do a excipient compatibility studies, and we did that. All types of excipients we add to the material, and we can check. You can see it is with sodium propyl paraben, sodium methyl paraben, magnesium stearate, tack, and microcrystalline crystal lactose, and we found that they are actually making it stable, and we are not losing anything. So we went ahead further for doing the formulation and trying to treat the animal models. Next. So a bioactivity confirmation is required, because whatever our purpose is to protection of the neurons, rat cortical neurons, we did in vitro and in vivo experiments to show that. Next. So we did in vitro studies by taking the primary rat cortical neurons and they are cultured for 10 days, then followed by 1 hour OGD, means oxygen glucose deprivation. When the neural cells are deprived from the oxygen and glucose, then naturally they will try to dry, and that is the condition where you will add the our material and then check with the two markers. One is dendritic marker, and then other one is the mature neuronal nucleus marker. That is, dendritic marker is MAP2, and the new one is the mature neuronal marker, so that you can find it out actually whether there is any improvement or not. And the pictures you can see in control and OGD, you can see the controls. They have the cells which are actually a lot of survived cells, whereas in the OGD cells, the cells numbers are less. And if you put in this graph, you can find it out. The OGD actually significantly decreased because of the oxygen deprivation. Next slide. Similarly, we checked actually whether these markers, whether all these 5 markers which are we have taken few markers and checked whether this improvement is there after in the OGD model, and we can find out in the wiedanolide 8 and 10 micromole. You can find out there are comparable to control. It is better than the only OGD model where the neurons are found to be dead. So that is why wiedanolide A appears to protect the OGD-induced dendritic shrinkage and neuronal death. So repeat experiments to quality the extent of neuroprotection is in progress, and we have 3 sets we have made already, and we found the similar results again and again. Next slide. Similarly, we also checked with wiedanoside 4, another marker, and you can see from the OGD vehicle treated, if you compare with the wiedanoside 4, the improvement is around 2 times, more than 2 times. That means actually this material actually working for neuroprotection. That is our AF1, Nimitli AF1. Next. We also checked with another marker, that is wiedanoside 5. There also we found more than 2 times of improvement in the neuronal cell, in protecting the neural cells, than in the OGD cells. So we are confirmed that in vitro condition, this wiedanoside 4 or wiedanolide A, both of the markers, they are actually protecting the neurons. All the 3 markers were tested, all them are protecting the neurons then in the OGD cells after they are added to the in vitro system. Next. So then we took this is mechanism of action. We studied actually this how this is working. Perhaps it is working through ROS pathway. The reactive oxygen species they are actually suppressing this things so that further neuronal damages are not being done. Next. So we did also in a model, in the stroke model, actually there are not many stroke models, but we have MCO model, which is nothing but a rat model where mid-cerebral artery is being blocked and oxygen supply is stopped for some time, for 2 hours, and then the reperfusion is allowed, and after reperfusion we start treating the 118F1, and we can able to observe that this material is actually working for protecting the neuronal cells. Next. So this is an MCO model. All of you must be knowing how it is working. Those people who are working in the reading in pharmacology, they might have used this model. And we could able to see that in 50 mg per kg amount, if you are treating the rats with immediately 118F1, it is giving the most ideal way of protecting the neurons. Next. So these are some of the brain pictures you can see in the MCO models. The brain is being disturbed after the blockage of the mid-cerebral artery. But after treatment of the immediately 108F1, there is improvement, and lot of improvement. You can see the deoxygenated area is white area, whereas it comes back to the red area where blood supply is there when it is treated with 118F1. So that shows that the 118F1 treatment actually protect the neuronal component of the brain, so that the oxide lack of oxygen is actually not damaging the brain. Next. You can see the neurological scores we also did in the same animals, so that you can see the neurological MCO model. It is around 8. Actually, in 10 number, we 1 to 10 we score, we observe the scores. So it is more than 8 damage. But when we treat with this, then we have got actually 56.72 improvement. So that is a good thing we observed. And it is in fact volume also. What is that area of the damage of the brain? One can see that the limitedly treated animals, as I have showed in the previous picture, the improvement comparable, very much improved than the in fact volume which is shown in MCO model. So actually MCO model is around 70% damage, whereas the in fact volume in the limitedly 118F treated ashwagandha treated animals, it is little more than 20 plus. So if we are thinking, because the model has got limitations of 24 hours, if some other model we are trying to find out can be shown that it goes further improvement. So that is the conclusion we have, that administration of this material ashwagandha limitedly 100F1 provides neuroprotective effects. Then the brain, okay, and then the MCO models which is not treated with anything. So within the 3 hours, if you are treating, so you are getting the improvement. Next. Then of course, as I have told, that we have to show the pharmacokinetics, and this material we did the pharmacokinetic study. Hello? Hello?
No, sir, you can continue, sir.
Okay, so pharmacokinetics we did. Next slide. So you can see all these 5 markers. So we showed that the material is up to 8 hours. It can stay in the plasma, and all these 4 we actually while applying IND, we will give the best one, maybe with anolide A or with aferin A, so that only 1 marker they want, although they require 4 markers. They may be biologically active or chemical active. So we will give 1 marker which is biological active. That may be that will suffice our requirement, and of course others are chemical markers. Next. Similarly, we did this the detailing Cmax, then AUC and Tmax and THAP. You can find out the all these markers what we have studied and how they are available in the body, in the plasma, in the rat, in 50 mg per kg doses. Next slide. Similarly, we did in the brain, because anybody will ask this question. You are developing it for stroke treatment, for protection of the neuronal cells. So it has to go to brain. You have to show it in the brain. So we showed it that in brain also it is working perfectly, and they are available. And in nanogram concentration, all these 5 materials are available in the brain. Next slide. The detaining numerical things are also there. You can find out the Tmax, Cmax is 4.04, Udanolide is 4.56, Udanolide B is 6.46, and Udaferin A is 6.65. So all these are available in the brain tissue also after the treatment, after 24 hours. Next. Then once we confirmed it, next slide, and this is just a consultation we did, and we time to time consult with the doctors, because we wish to see that whether it can be actually useful or not. So they have given the positive remark that if it is possible to expose the animals to the phytopharma during the ischemia, so the moment the ischemic condition is there, if you can feed and so it so that animals the acceptance will be more, and the earlier possible intervention can be, if you can be shown in the experiment, so that it can be planned accordingly for the human treatment. Next. After knowing that 50 mg per kg body weight can work in rat, so that according to that dose we calculated, and it may be the in human it will be around 500 mg as per the formula, that body surface area calculation. So we saw, then we did actually survey whether it can be acceptable in the market or not. You can see the global ashwagandha market is actually growing and growing and growing, and by 2029 in it will be around 102 US dollar million US dollar in Asia, Africa, America, and Europe, all the continents they have. And this is a good market globally. So we we are very happy that we can actually project this material phytopharmaceutical for the treatment of the stroke. And the stroke molecules of similar types are not available, and the phytopharmaceutical is certainly not there, because it's a new area altogether, and there will be a commercial things which are actually new companies are coming up which can actually develop the phytopharmaceutical to help the scientist. Next slide. Next. So all these things, toxicity studies we did. Next. So as the regulations require, we do 2 single dose acute toxicity study in rat and mice, and we found that 2 gram per kg body weight of this material can do not do anything to the animals. So it's a great window actually to show that the material is very, very safe, and the 50 mg efficacy dose, whereas 2 gram is not adverse. So that is the maximum dose actually DCGI or FDA requires. So we could able to confidently tell that this is going to be a very good one if it is showing some efficacy in humans. Then we did a repeat dose toxicity. Next slide. So in repeat dose toxicity, we did actually for 28 days because we wanted to put this drug as early as possible in the market. So 28 days repeat dose toxicity studies we have done, and we did a reversal study also, because whether if there is any changes, the reversal animals, whether it is getting reversed or not. So we found out that there is no effect on alertness, activity, behavior, fur coat, discharge of any kind from the body, and diarrhea type things and sickness. All these things are observed during the 28 days of period. Then we observed actually 20 the reversal for 14 days, and we could find out there is the phytopharma is very, very safe in the rat at least, so that we can go ahead. Now we are actually planning next. Now we are actually planning to do it in the different animal model, and after that we'll go to a human system. We will file the IND and check for human studies. Next. So that is what I have to say today. I hope you people understood something. Any doubt, any slides, any place, please be open and tell me where I can explain you again the things. Thank you so much.
Bhavna ma'am.
Thank you so much, sir. If anybody has any questions, you can please write it in the chat box, or you can unmute yourself and ask the questions.
Good afternoon, sir. Good morning, sir.
Good morning.
Good morning, sir. Sir, one of the important things that for stroke, actually signal to the neuron certainly it receive very high intensity signal, spark is very high. Sometimes brain stroke is happening. Now, before that, if we can minimize this stress in the body, there is any medicine, these types of thing is there. Is all types of medicine is working similarly, because man to man states level are different, and different people have different types of states also.
Actually, problem is that of course, as you said, that all of us are very, very different, and our lifestyle and physiology is very different. Okay, but while developing a drug as of today, of course not pharmacogenetics. The classical way of developing a drug is that during the clinical trials, maximum number of the samples are being checked, and on the basis of the observations, we actually give the drug a permission for use in the majority of people, right? This drug, what we are developing is not actually a clot buster. It is actually providing some protection if there is clot is happening. Okay, that is what is. And the problem is that while somebody is suffering from stroke, it is very, very accidental. Okay, all of a sudden it comes. Nobody knows when there will be a stroke, right? So in that condition, actually, it is very difficult to treat the patient before anything, before the stroke happens. But if stroke happens, then such type of phytopharmaceutical can be utilized to minimize the damage to the brain. That is what the our aim, to minimize the damage to the brain.
One thing is that if there is some pre-symptom is there for stroke, sometimes 70% there is some symptom.
Yeah, that is true. If there is a pre-symptom, for example, debility, okay,
Yes,
somebody may be restless, feeling something very different abnormally,
heart rate is
right? So in that time, it can be, this can be treated.
That's
That's what. this our we are positive enough, thinking positive that it may work, but still we have to prove that it works like that in the phase 2 clinical trial or phase 3 clinical trial.
Okay, one of the important things, sir, when you are testing this medicine,
Right.
some probabilistic model you are using this level of acceptance, this level of rejection. Sometimes I am from computer science background. We
Right.
are using false alarm, false accept, false reject. But there's a
Right.
high chance
True,
of
true, true.
true. Now, these types of testing, actually, probabilistic model we are using here for when you are accepting or rejecting.
Exactly. Actually, we accept and reject on the basis of efficacy. For example, this drug we tested, candidate drug, I would say, we checked in different doses and we found there is a difference in dose response. Okay. So there is a dose response curve, and on the basis of that, we actually decided that 50 mg per kg body weight the dose, which can be the producing the best result. Okay. So on that basis, and our limitation is we do not have many more models which can actually be experienced. So if many more models are there, certainly we can test in different models and check whether it is working in many models or only one model or two models. So this model is actually mice and rat model, MCO model. And rat is a better model than the mouse. And we checked that in a different doses, dose response is there. That means it has some efficacy. That is the confidence level we use for pushing this candidate further.
So in that case, you are changing the parameter values for adapt these situations to situations.
Yeah, actually that has to be checked. No? That has to be every parameter should be constant for all the models.
No, there should be some range, flexible range. Within that range, you have to optimize the value.
Exactly. We did that. I only showed that the most successful story I showed. Otherwise, we did these things in different doses. We checked actually 5 doses, 10 to 100 we went.
200. Actually, if I'm testing for more than 30, that Gaussian types of things, and in natural way it is coming. Even large number of samples are considered and testing.
Right.
With the
Yes.
good result.
So that is again I would say we are actually due to the ethics rules, we don't have to cross number of animals used for a experiment, for particular experiments. So nowadays government of India tells the minimum animals you should use to derive the most logical scientific evidences. Otherwise it can be explored in different conditions, and one can find out many different results and compare actually what you are doing in silico. These are some of the limits nowadays in vivo studies.
Therefore, when you're plotting the graphs, all these things, which types of you are writing, your group is writing code, or you are using any software? There's a lots of graphs you are showing.
Yeah. As softwares we have when you are talking one slide I showed, right?
Yes, yes.
Neurological deficit. There we have to
Yes.
observe under the software. Actually, they are inbuilt computer-based platforms, and you can with different cameras and other things, the animals' activity and other things being observed. Then it gives a score.
So what's the name of that software? It is available with your
that is actually, if you are buying that particular software, I can tell you we have, and there you have to one has to buy that with the total entire system.
Okay.
So they are patented, and only because the purpose is goal is only one goal, that is the assessment of this. So they are not commonly available.
Any sir open source software is there?
No, sir. Actually, open source software people, they don't do because use is not there. No? Only specialized people will use that. For example, PCR primer software, designing software is very common, right?
PCR
But
primer.
right, for these type of softwares, very little is there.
Your graph is very good, neat and clean. That's what I'm asking.
Sir?
When your graph, whatever graph we are presenting, it is very
Right.
neat and clean. When you are
Right.
writing code and we are plotting, actually by using some our operating system, it is not so fine. Fine. Okay, sir. I am from computer science background. I am computer science faculty. I am very interested because I am working some area of brain neurocomputing, brain signaling, signal handling.
Okay, sir.
Okay, I'll contact with you.
Yeah. Sometimes you can visit our drug research institute. We have a big neurology department, so you can interact with us and we can plan better.
This is a very bigger area. State, strain, brain, neurocomputing, brain.
Absolutely.
Okay, this is emerging area. Thank you, sir. Very nice presentations.
Welcome.
Thank you, sir. Thank you, sir.
Thank you, Dr. Shrikant Rath, for enlightening us on the recent developments in the field of cytopharmaceuticals. And our next speaker will join us at 12:30. So let's take a very quick tea break and rejoin the session, or stay connected in the same session. By 12:25, our next speaker will speak on recent trends in the field of humanities. Thank you so much, sir.
Okay. Thank you, madam.
Yeah, thank you once again, Srikant sir, for
Thank you.
accepting our invitation and give a very precise and informative research tools to us. So thank you once again, sir.
Welcome.
And very soon we will call you in our campus in offline.
I will come.
You can leave, sir. Yeah.
Okay. Bye.
Yes, sir. Thank you, sir.
A very good afternoon to all the dignitaries, faculty members, academicians, and research scholars from the various universities and institutes. I welcome you all back to the session. We had the first session with Dr. Shrikant, where he gave us insights about the recent trends in phytopharmaceuticals. In our next session, we have Dr. Fayaz Ahmed, who will be enlightening us in the research in the field of humanities. Before we start, I would like to read a very brief about Dr. Fayaz. Dr. Fayaz is the assistant professor and local head in the Department of Studies and Research in English at Rani Channamanna University, Karnataka. He is an MA and a PhD from the Karnataka University. He has a teaching experience of 9 years as an assistant professor and is a research supervisor, and he has 4 students under him. He's guiding 4 research scholars, and his projects have been post-diaspora multicultural society in Zadia Smith's select novels, and an ongoing project in digital tools as an aid for research scholars. Sir has published numerous papers. Some of them, Sir has published 21 research papers in the national and international conferences, and he has published 1 book entitled Family Life and Society in Select Plays of Arthur Miller. Sir has recorded 13 online lectures on research methodology for MHRD, Government of India, Ramanujan College, and Pandit Deen Dayal Teaching and Learning Center, New Delhi. Sir has over 903 subscribers in his YouTube channel. He has been invited to deliver talks as a keynote speaker plenary sessions, and he has chaired sessions at national and international conferences. Sir is working as the local head in the Department of English, RCU PG Center, Vijaypur. He is a member of the sports committee and is also working as a coordinator for minority cell. Sir is a member of several organizations and he is in the board of examinations of various institutes. Sir is a member of Indian Association for Comparative Studies and is also a member for the English Studies Circle South Africa. We welcome you, sir. Over to you, sir.
Yeah, thank you, Dr. Bhavna ma'am, for such a sweet introduction of me. And then I take this opportunity to thank the Amity University for to act and just express my views as a resource person in this 5-day faculty development program, which is on recent trends and challenges in research. I don't know how much I will be able to fit in your ideas, and then whether I will be able to provide you justice or not, but then also it will be my humble attempt in this regard. And good morning to all the members over here, the executive members, Dr. Jayita Chattopadhyay, Dr. Somen Kandra, program convener, Dr. Amit Kumar Datta, and my dear friend, Dr. Shantini Banerjee, and then the FDP coordinator, Dr. Dhananjay Pandey. The topic when Shantini told me that you need to speak on copyright and publication, and she insisted me to present it from humanities perspective. But I have my own reservations, but I will just make an honest attempt. How much I will be able to fulfill or fit in those scheme, let's see. But still, I have made an honest attempt in that regard. But before going to copyright and publication, it's important for us to understand what is intellectual property rights, because it is one of the copyright is one of the component of IPR. IPR is nothing but intellectual property rights, as we all know. And this is a term which is reserved for types of property. I hope I'm audible, right?
Yes, sir, you're.
Yeah. Thank you. So this IPR, that is intellectual property rights, is growing in importance, especially since the coming of existence of TRIPS and its implementation in India. And also the advances in digital communication and technologies have been major factor. The term property generally brings to mind two types of properties. One is immovable property, for example, land, buildings, and movable property such as jewelry, shares, stocks, automobiles, etc. Here, intellectual property can be considered a third category of property. So intellectual property can be considered a third category of property since it has been increasingly being recognized as of prime importance in the economy of nations of any country it may be. And the basic feature that apply to other types of property, that the owner of the property is free to use as she wishes, provided the use is not against the law, and to exclude other from so using that own property apply just as well to intellectual property. Is it fine if I don't enable my video since there is no power? I may run short.
Okay, sir, no issues.
Yeah. Just a minute. Yeah, sorry for the description. So here, what exactly is intellectual property? The term is reserved for types of property that result from creations of human mind, that is the intellect. Intellectual itself means the intellect, and these property rights, they ensure that it is being protected, and the rights of ownership are granted by the state. However, unlike the conventional types, the rights of the owner are limited to a specific period of time, which I will be discussing in my later slides also. So the categories of intellectual property goes like this: trademarks, there is marks, logos, names, etc. And then the patent, invention, for example, a new product or process of manufacture. And then industrial designs, for example, a new shape for a bottle, so on and so forth. And then copyright and copyrights related rights, literary, artistic, and scientific works, books, etc., performances, broadcasts, etc. And then the last one is geographical indications, geographical names indicating a product. So all these are covered in, these are the categories of intellectual property. So now, who looks over it? In India, it comes under the control of the General of Patents, Designs and Trademarks, which has been administered all matters pertaining to patents, designs, trademarks, and geographical indications. All these things come under the Controller General of Patents. And the Indian Patent Office, functioning under the Control General, is also recognized as an international searching authority. And then there is one, the Rajiv Gandhi National Institute of Intellectual Property Management, which is affiliated or comes under the Ministry of Commerce and Industry. And the link is provided over there. I will not going to click on the link. And then the copyright is administered by the Copyright Office under the Ministry of Human Resource Development. So those who do not have any idea about the copyright, there is an official website. There's an official website wherein you can go and find out all the details if you at all want to know, or if you want to apply for copyright process. And then what is copyright? As per the standard definition of Oxford English Dictionary, the exclusive right given by law for a certain term of years to an author, composer, etc., or his assignee to print, publish, and sell copies of his original work. This is the standard definition which I have picked. There are a number of quotations and definitions which you find. So now, the basic question is why we need copyright. As I told in the earlier slides, I have discussed about intellectual rights, and this comes under this. So it ensures a fair play to the author as well as to the reader. Here, the reward of creative efforts is being ensured so that the author should feel that his work, thou shall not steal. So no one can steal his intellectual property. And then he will be having exclusive rights for a limited period, of course. And then copyright is very, very essential in order to promote research and in order to promote creativity. So once if you feel that there is no protection for your works, then there will be no work at all. So in copyright, what are the things which can be protected and what are the things which come under it? First one is literary works. So it may be in any form, whether it may be novel, drama, prose, poetry, whatever it may be. And then dramatic works also, it may be a movie or a documentary, so on and so forth. And then photographs, paintings, motion pictures, TV programs, architectural and other artifacts, software, databases, sound recordings, and so on. So this is how copyright works in national versus international. Remember, there is no single copyright or international copyright for the globe. It changes from country to country, from globe to globe. And the Copyright Act of each country is compliant with most international conventions and treaties, as in how there was a need and demand. There has been some amendments made in the international conventions also. And then in India specifically, the act was introduced in 1957, or it came into effect in 1957, and is compliant with these international conventions as well, and treaties also. The more prominent or the very important convention and treaties are the Berne Convention of 1886, and then the Universal Copyright Convention of 1951, and the third one, Agreement on Trade-Related Aspects of Intellectual Property Rights. So to speak about from Indian perspective, as I told you earlier, this first act came into being in 1914, followed by the Copyright Act in independent India in the year 1957. It created Copyright Office and Copyright Board to ensure that these activities are carried out smoothly, and introduced civil and criminal remedies against infringement of copyright, which I will be discussing at length in my coming slides. And then copyright protection are in two forms. One is economic, the other is moral right. So here, economic right, which gives us some monetary benefits. It gives several exclusive rights as follows: to produce copies or reproductions of the work and to sell those copies, any number of copies, to import or export the work from local to from regional to national, and from national to international, and then to create derivative works that adapt the original work also, and then to perform or display the work publicly. Just a minute, there is some disturbance over here. I will step down just a minute. Sorry for the inconvenience caused. Yeah. So this economic right ensures to perform or display the work publicly also, and then to sell or assign these rights to others. So it will be the owner of the copyright who will be having all these privileges, and he can also earn out of it. And then to transmit or display by radio or video or any form of media. And then when it comes to moral right, he will be having two moral rights. One is right to paternity and right to integrity. So here, right to paternity is to claim authorship of work and to prevent all others from claiming authorship of his work. So it will be exclusively his own. And then the right to integrity is to prevent distortion and then mutilation of or other alterations of his work or any other action in relation to said work, duplication or using it for monetary benefits without permission. And then here, this right to integrity prevents such action in relation to said work, which should be prejudicial to his honor or reputation. So the term of copyright or the duration of copyright, it differs from country to country. So here, the duration of copyrights varies from country to country. The minimum duration of copyright protection under Berne Convention is 50 years. In the European Union and in the US, the duration in author's life plus an additional 70 years. In India, copyrights are protected for a period of 60 years. So you can find the difference over there. And then from the beginning of the calendar year, next following the year in which the author dies. In the case of original literary, dramatic, musical, and artistic works, the 60-year period is counted from the year following the death of the author. And in the case of cinematograph films, sound recordings, posthumous publications, anonymous and pseudonymous publications, works of government, and works of international organizations, the 60-year period is counted from the date of publication. Though it is in brief, and this is how the copyright registration flowchart works. You have to go to that official website, and then there is one particular format, and you have to fill the application along with fee. And you can even attach a DD, that is demand draft. And after filling all those details, there will be one number which they call it as diary number, which will be issued to the applicant who wants to copyright his work. And then there is a 30 days mandatory waiting for objections. And you can see there, if objections are filed, yes or no. On the left side, if there is no objection, it goes to scrutinization by examiner. And then if there are discrepancies found, it goes the discrepancy letter issued to applicant, and then the reply from applicant and hearing by the registrar due to rejection. If there is no objection or objection filed, sending letter to both parties. A letter is being sent to both the parties, the one who has applied for copyright and the one who has filed an objection to give copyright to that person. So after that, the office collects replies from both the parties, and it is being heard by the registrar, and then the decision is being taken whether the application is accepted or rejected. And then the next process goes on. If application is rejected, application gets rejected, sending rejection letter to the applicant. And then if it is approved, registration is approved by the deputy registrar, and then sending extracts from registrar to the applicant, and then the copyright will be issued to the applicant. So what are the benefits of copyright? Here, I have pointed out some 7 benefits of copyright. One is the legal protection, so that there cannot be duplication of your work and unauthorized. He cannot use it for other purpose without a due permission from the original author. It happens so knowingly or unknowingly, we just fall prey to such things. So it's better to copyright, whether it may be your book or your article or whatever it may. And then it also ensures that there is a brand value given to your work. And this copyright also ensures that you have worldwide protection for your work, and then the copyright becomes your asset. And it also ensures that your work is being publicized by the name of the author, but not by the name of the publisher. And then there is economic stability also, which you get by registering for copyright. So now, how to ensure that there has been no counterfeiting to copyright? First, the counterfeit mark, the production of certain brand bags by putting a false label and sold at low prices. In the market, you must have seen so many such false labels. For example, Nike, Nike, that is the registered copyright, but they just use it, they change it, and they change the spelling, they change the model and rename it and sell it in the market. And then, for example, even copies of articles similar to the original under a different name, that is also liable to be counterfeited. And then infringement mixed, copy for both the model and brand. That is, for example, fake Louis Vuitton bags or Rolex watch, so on and so forth. Infringement of copyright, copying or using the work of another creator and naming that it is yours without his prior permission. So now, what are the remedies for copyright infringement? So there are remedies in 3 levels. One is a criminal, the second is, first is civil, the second is criminal, and third is administrative. Civil is non-punishable act, and then criminal is a punishable act, and administrative, which you can see it on the official website of copyright. There you can just address and express your grievance over there. So criminal offense, if done unintentionally also, you are liable to imprisonment of 6 months or 3 years. And then fine goes up to 50,000 to 2 lakh rupees. And also there will be seizure of infringing copies as well. So it is under the section 63 to 69. And in civil remedies jurisdiction, it is heard in district court under section 54 to 62. There is injunction, damages, accounts, delivery of infringing copy, damages for conversion, so on and so forth. There are limitations to copyright as well. Idea expression dichotomy. So it happens so many times, especially those who are pursuing their research, they fall under this trap, wherein they have due credit to the author, they have cited, but then also there is some idea of the previous author or the previous research scholar, which has been again expressed over here and which falls under the category of plagiarism. So the work here, the original work gets covered in copyright, but it's very hard to protect the original expression of ideas. So what can be done here? It cannot control the idea which is being presented in a new perspective to the existing idea which was there earlier. So this is where the limitation of copyright comes. And especially it happens in movies and movie scripts as well. And then the first sale doctrine does not restrict the owner of a copy from selling legitimately obtained copies of copyrighted works. So this is a double-edged sword, what I believe. While signing the contract, one should be very much aware as to the publisher is getting signed for how many number of copies. And sometimes it goes unnoticed that he gets it signed for 100 or 500 copies, but he goes on to say publish more than 1,000 or 5,000 or more than the number which he has assigned in the contract. Sometimes it's very difficult to find out or locate. So that's why this first sale doctrine, copyright has limitations over here as well. And then the third one is fair use and fair dealing. So most of the books are used for the purpose of research or private study, learning and teaching in a classroom, criticism or overview, reporting current events for judicial proceedings, performance to non-paying audience, and other specific conditions there, the copyright cannot cover. So moving on to publications, we can identify that there are 12 kinds of publications. The first one is book notes announcements, that is editor refereed. Second one is newsletters and newspaper articles, that is again editor refereed. Reviews, continuing commentaries, forums, and then conference papers, which are both refereed and non-refereed. Journal articles, refereed, single blind or double blind. Journal articles, those are editor refereed. And then chapters in books, solicited, commissioned, and referred. Books, scholarly, editor reviewed and refereed. Textbooks, editor reviewed and refereed. And then the last one, other property, self-publication, etc., vanity publications. So you can just have a glance at the publication process, how this publication process takes place. In order to assure quality in publication process, authors, editors, reviewers, publishers, all are involved into it. First, when you submit your article, there are some requirements based on international standards. You need to, after submission, you need to revise, you need to recontend, and you should also follow certain ethics which are prerequisite. And then it goes to peer review. Peer review are again blind or open. And then editorial decision and input. There can be reasons for rejection. Revision include plagiarism, and then there may be redundancy, substandard research, faulty or incomplete analysis, poor or incomplete reporting. So the entire right rests with the editor whether to accept or reject your research paper. And then if it is accepted, it goes for revision. They ask you to revise certain things, certain elements, and in order to improve the quality of the paper. And then it goes for production, and then post publication scrutiny is also taken place. The author submits to a journal, and then the editor screens it. Editor invites reviewers, and then reviewers score and suggest decision. Revision and amendments. Editor reviews and sends decision feedback to the author. So here, there are types of peer review. One is the single blind and the double blind open peer review. So what is this single blind or a single anonymous peer review? Here, the reviewers know the identity of the authors. The identity of the author is being revealed to the reviewers, and it is been sent by the editor of the journal. So here, authors do not know the identity of the reviewers after submission of their articles or research papers. And most common model of peer review is STM. And then the double blind, double anonymous. Here, the reviewers do not know the identity of the authors. So in the previous single blind, there the reviewers knew the identity of the authors. But here, wherein in double blind, the identity is not revealed. Authors do not know the identity of the reviewers. Most common is HSS. And then in open peer review, reviewers know the identity of the authors. Authors know the identity of the reviewers. Reviewer reports may be published with reviewer names if article accepted. So in my teaching career, I have come across this problem as to how to find out a right journal. Most of the budding research scholars as well as the faculty members, since there have been so many amendments by the UGC time and again in order to ensure zero tolerance to copy paste and plagiarism. Also, there are 2-3 softwares which are there in the market to ensure that due credit is being given to the original author. So here, finding out right journal is a task. Is a very difficult task. So certain things which you can consider is the type of journal in which you are sending your research paper, in which you would like to publish. Check the references in your article, especially in the introduction section, to give an indication of possible journals of interest. So you may be just a moment. And then read the journal's aims and scope on the journal homepage. Read or download the journal's guide of authors. Check if the journal is invitation only. Some journals only accept articles after inviting the author to submit. If you send them directly, they will not accept it, or they will just reject them. And then check the journal's performance for review and publication timelines. If you need to publish open access, remember the most journals explain their open access options on the journal homepage itself. And then submit your paper to only one journal at a time. So there are certain journal metrics which you should consider and which you should take a note of it. Journal metrics help you to select the most appropriate journal for your article. So You can also identify through the keywords, which I will be telling you with an example in the coming slide. And then alongside the journal's scope, editorial board, international outlook, and audience, help find the best destination for your research. And there are different types of journal metrics. You have to look at more than one metric to help you make your decision. You will find a dedicated journal insights section on many of journal home pages, giving information about the journal's speed and means when you have to submit, and after submission, how much time they require to review your paper, and then when it will be online, when it will be published online. So all these details you have to ensure. And then reach your article. Once if your article is published in that particular journal, what will be the outreach of the readers? That should also be kept in mind. Geographical location of corresponding authors and journal usage is also very important. That's why this impact factor journals play a vital role. And an impact metrics based on citations received by articles is also very important for you to consider. So here, one more suggestion which I would like to give you is that there are number of, this is a new way of finding out as to where you have to publish your journal. So this is called as Journal Suggester tool. The link is provided here. And this is how it works. So almost most of the standard journals like Springer, Scopus, and then SCI journals, you just type in the Google search engine, Journal Suggester tool. There is a Springer Journal Suggester, Taylor Francis Journal Suggester, and then Elsevier is there, LibGuides is there. So just let us click on one and find out how it works. Yeah. I will copy paste my abstract, a research paper of mine which is not published yet. Copy this. Put it here. The manuscript title. So now subject area, from which background or from which field you are. You have to select there. Since I'm from here, humanities, English literature, and the paper is from one novel written by Patrick White, and the title is The Solid Mandala. So literature. And then now I click. If you want to refine your recommendations, you can refine. Here you can give minimum impact factor, and then minimum acceptance rate, maximum time to first decision, and then indexing service. And then again, option choice is given there, whether you want to publish it in any kind of journal or open access journal or subscription journals. So let us go for open access journal, because here the yeah. And now I click for no journals found matching the search criteria. Okay, fine. So let us go for all journals. Yeah. So there is one journal and the impact factor of this journal is 0.2. And the first, this is 70 days average they take. And that is the acceptance rate of this journal. So since my topic is a limited one, hence I have got only one suggestion. So it depends on the nature or the topic. Even in order to boost your recommendation, you can use keywords as well. Here, I have not used keywords. Yeah, you can include it here. For example, disability, sorry. Yeah. And then social paradigm. And now let's see how many recommendations we get. But unfortunately, I couldn't. But it depends on which keywords you use and in which area you are sending your paper. And let us try with one more paper. So let me copy paste the title of the paper.
Fayaz sir, Dr. Dhananjay this side. Sorry to interrupt you.
Yes, sir.
Actually, we can see just the slide journal suggested tool link. If you are trying to show us something else, we are unable to see, unfortunately. We are on journal suggested tool, the slide, last slide, where you have shown the link, google.com link.
Yeah.
Yeah, thereafter, if you are
So-
trying to show somewhere, means like I think you are trying to show us the web page where you are pasting links, abstract or something else like that.
Mhm.
But unfortunately, that is not visible to us.
Is it? Oh, I'm sorry for. I'm so sorry for that.
No problem, sir. No problem. I think you have to share the screen for that.
Yeah, already it has been shared, sir. So, is this Google search engine? Is it visible to you?
No, we are on the slide, sir. The slides what you are showing, journal suggested tool, the last slide what you left.
Now,
No, sir.
my screen sharing is paused from your side. I think so. Someone, just a minute. Yeah. Mhm. Let me try once again.
Yeah, please.
Now,
Yeah, it's coming, sir. But it's still
is it
slide.
visible?
Slide. Yeah, we can see.
Now,
Yeah.
okay. So, if you put it in the search engine in Google, you can the journal suggested tool. You can find number of Springer and Scopus journals, SCI journals, and this is For example, let's see this Springer Journal Suggester. And then here, this is my paper and the title, which I'll copy and paste it, manuscript title. And then in manuscript text, you need to just put the abstract. No need to put the entire paper. And you can see in this article also I have not put keywords, but I suggest you to put keywords so that. Yes, ma'am, got it. Yeah, I did, ma'am. I did so. Yeah, Dhananjay sir, is it visible?
Sir, still we can see slide. I think you have to stop the sharing of slide and you have to share a screen. There will be the option, the window, sharing of window or sharing of screen. So I think when you will share the screen, then I think we can we will be able to see. Maqsood sir, are you here, sir? Maqsood ji?
Then now I have stopped it.
Yeah. Then I think you should share screen.
Yeah.
Now
Now,
you can see.
okay,
Yeah.
fine.
Yes,
So
sir.
you can see there. Yeah,
Yeah.
in manuscript title, I have put in the title of my research paper. And then in manuscript, you need to paste only the abstract. No need to put the entire article. And I insist you to use the keywords there so that more number of journals are covered into it. And then, as I told you earlier, here in this box, there are subject areas, means it covers almost all the subjects. Here you have to select from which subject area you are. And since I am from humanities and my is literature. So here, minimum impact factor in whichever journal you want to, you just put the number over there, and then minimum acceptance rate 15 days, 1 month, 2 months, 6 months, like this. And then maximum time to first decision, means how long you can wait for the acceptance of your research paper. And then indexing is also a new criteria nowadays. And then here, as you can see, view, you can select all journals or open access journals or subscription journals. So let us go for all journals here. And then go for click suggest journals. And unfortunately in Springer on my topic, there is only one journal. But in other subjects, it may differ. So let us try in Taylor and Francis journal suggester. Here again, it is the same way. I have already displayed it. There are two easy steps. Step one, paste in the full abstract of your article. The suggestion will be more accurate if you use a full abstract containing relevant keywords, as I had suggested you previously. And then step two, click on reveal suggested journals to see a short description of the journal and some citation and speed metrics. So here there is only one column wherein you can put the title and the abstract together. Yeah. So you can see there, the title is there and the abstract. It doesn't matter whether you mention abstract or not. Or else let me type it. And then now let us go for reveal suggested journals. Yeah, you can see now here there are number of journals which come under Routledge publications. Here there are some suggested journals like Asian Ethnicity, Contemporary South Asia, South Asian Diaspora, India Review, South Asia Journal of South Asian Studies, Social Identities, South Asian History and Culture, South Asian Review. There are so plenty, a handful of journals which can accept my research paper. I hope I'm clear with this concept. And then again, going back to the main page wherein we had tried to search journal suggested tool. Here you can see there are different types of publishers, and whichever, even elsewhere also it's there, and whichever you like to publish your journal, you can try and experiment with it. And even here also you can see that almost most of the publishers have this journal finder tools like Web of Science and then Scopus, Directory of Open Access Journals, EndNote, Elsevier, Open Journal Matcher, so on and so forth. Yeah. So now coming to UGC care list. Most of the research scholars and faculty members from India, they are concerned about this. I think, am I sharing my screen or no?
Yes,
Yes.
sir.
Can you see my slide?
Yes, sir. No. Not the slide. We
No,
can see the
yeah, again it
screen.
is asking for resume share. Just a minute. Again, once again, I will share it. Now you can see my slide, right?
Yes, sir. Yes, sir.
Yeah, okay. Thank you. Yeah. So now this is an area of concern for most of the research scholars as well as the teachers from India. And the government of India has been ensuring to promote research, and then let there be transparency as much as possible. Earlier, they called it as UGC approved journals. There were so many journals, and then since they were involved into some other things, so again they filtered some of the journals and they called it as UGC care list journals. And this UGC care list journals are again divided into two categories, that is list 1 and list 2. So now, what's on the display?
It's web page, sir. Right now
Yeah,
we are
page, right. Yeah. Thank you. So if I type here in the Google search engine as to UGC care list, it leads us to the official web page of UGC, which itself is called as UGC Care, which is being taken care by Pune University. So here, what you have to do, the prerequisite is that you have to log in or if I, of course, I have registered for it. And if you have not registered for it, you have to create an account and then you have to log in over there. Yeah. So this is the page interface which you come across once if you log in there. And then you can see that there has been an alert. There has been a flag raised over there. There are certain cloned journals in both group 1 and group 2. Before sending your article, please ensure that those journals are not cloned and they won't fall in this category either. It may be in group 1 or group 2. And here UGC care list group 1 search engine is there. There you can search by title, by ISSN number, by publisher, by subject, and language of publication. So let's see here, the subject, let me put English, and then let's go for search. So it asks to log in. If you don't have an account, it will ask you to create an account and then you have to log in. For that, it is free. You need not pay anything for it. And then here, you can see sciences is there, social sciences is there, arts and humanities, multidisciplinary, and Indian languages. Here, let me go by subject. English, search. So here, there is one journal which falls in UGCK list group 1. You can see serial number, journal title, publisher, ISSN number, EISSN. EISSN means the soft copy ISSN number. And then UGC care coverage years. See, my dear participants, you should be very careful to know at what time when you submit your paper for publication, whether at that time it is under UGC care or not. Certain journals have been discontinued. And see here, if you are going to send your paper in this journal, which has been already discontinued in the year 2022, April 2022. So now, if you submit, it will be under cloned journal. So ensure that it is still active before sending your paper to that particular journal. And now let us go to Group 2. Here the journals are part of UGCK list as Group 2 and searchable through following links. So Web of Science, as itself indicates, here for arts and humanities, there is limited scope, wherein there is many chances for science related journals to be accepted and published. And even there is a Scopus source list, which by clicking, you can just go through the list of the journals which come fall under this category. some idea about how this UGC list functions. There is lots of disturbance over here. I kindly excuse you that I'm going to cut short my lecture here itself. I thank the organizers for giving me an opportunity and to share some of my ideas on this topic. I'm sorry because there is some power disruption and a huge traffic. Heavy traffic is going on. People over here who are moving in and around because there is a lunch break now, and even my battery is draining a bit. Yeah, thank you once again. I hope I have tried to convince you and given some idea, some new perspective regarding the given topic.
Thank you so much, Dr. Fiaz. It was a very highly interesting and insightful session. We enjoyed how to find the journal. If you could, sir, please pass on the link, copy and paste it in the chat box, the journal finder. It will be very helpful for the researchers.
Yeah, sure, ma'am. I shall do that.
Yes, sir, in the chat box, please, sir. And anybody has any questions, you can please hurry up. You can please write it or you can.
Sir, in the UGC care list, there are many journals which are paid journals. So is it advisable to send papers to the UGC care list paid journals?
Yes, ma'am. It again depends on your requirement. Hello, am I audible, ma'am? Yeah.
Yes, sir. Once again, if you could repeat your answer.
Just a minute. Let me share the link first.
Okay, sir.
Yeah, I think I have shared it. Yes, ma'am. Kindly please repeat your question once again.
Sir, ma'am wanted to know that there are certain paid journals also under the UGC list. So for our students who are first-time researchers, is it advisable to send the papers there?
Yeah, in fact, I wanted to speak on this, but it would take some more time for me to throw light upon this. There are two types of journals. There is open access and commercial journals. So in commercial journals, what you'll be doing here, knowingly or unknowingly, you will be giving the copyright to the publisher, because you want to get your article published as early as possible. And without, he just bypasses, he doesn't send it to reviewers. There is no blind peer review, there is no kind of peer review at all. And then he gets it done through his, he spends money for the review committee and all. He pays them and gets your article published. But if at all you want to get your article, if you want to get your recognized as a good researcher, it's better, first thing what you do, ma'am, you create a research profile in Google Scholar or in ResearchGate and whatever articles and even academia.edu. And then there is news.edu also, I think. And so there, if you create your account there and put your paper there, so that your indexing and your citations will bring you some points and you will be recognized as a serious researcher. So better you go for the Scopus and Springer journals. As I told you in UGC care, again, there are two categories, list 1 and list 2. And in list 2, when it go in Scopus and other journals, they consume lots of time, and there is so much of process which you have to undergo. They test your patience. And if you are ready to get tested, better. I wish you all the best. Please go for that. But having said that, there is no point in getting 20, 30 articles in the span of 2 to 3 years. It's very fine if you have only 2 standard publications in 1 year, in 1 academic year. I hope I have answered your question, then.
Thank you so much. So it was a very insightful session. Thank you so much, sir. We learned many
Sorry,
important
ma'am,
things.
in fact, I am not convinced the way I have presented because I was nervous about this power fluctuation. This is
Don't
not
worry,
best-
sir. We'll invite you
this is
again.
Not my best. No, I'm not up to the mark. Even I am not satisfied with my presentation.
So we'll have you again in 6 months' time.
Yeah, thank
Because
you
we
so
loved
much.
what we heard from you and what we learned from you. Thank you so much.
Thank you so much. Thank you
I
so
would
much.
like to thank all the participants who joined today. And I request everybody to join tomorrow at 11 a.m., which is the last day of the FDP also. And please join, for those who are joining online, please join through the same link. And for those who are here offline, the valedictory session will commence in the seminar hall. So see you tomorrow at Amity University Chapel. Thank you.
Thank you, Fair sir, for accepting our invitation.
Oh, it's my pleasure, ma'am. Thank you so much for inviting me once again.
Thank you, sir. Thank you.
Thank you. Thank you, all of you. Bye-bye. Good day.
Bye, sir.